Hey, Dr. Baker, how are you?
Hello, I'm fine.
Good, good, good.
I was just getting ready
Ah,
to email you because I've been sitting in here for 10 minutes and nothing happened. I was worried
sorry.
I was in the wrong
Ye-
place.
No, you're in the right place. I joined a little late. I had another meeting
Okay.
and then had to run out and run a quick errand. So I just got back. So sorry about that.
No
Um,
worries.
just the advantages and disadvantages of working from home.
Of
But
course.
anyway, by all means, go ahead and start whenever you're ready.
Okay. It was unclear to me exactly who the audience was supposed to be for this. I've tried to make it as general as possible. Let's see. I need to be able
That's
to
perfect.
share my screen. Let's see. Can you see my screen now?
I can. Yes, sir.
Okay. I'm going to talk here today, tell a little story that starts back in about 1998, 99, actually, when I was working at UT Health Science Center. That kind of winds into what we're trying to do with this gap funding project. This really is going to be broken down into two stories. The first part kind of lays the background and describes the need that we have, and then the end will talk about what we're doing and how COVID has kind of impacted progress towards our goals. But basically, this first part is going to talk about our overall uh, interdisciplinary collaborative efforts at ligand discovery for G protein coupled receptors. And this is work that I've done in collaboration with Dr. Abby Perel, who is both a professor in the Department of Chemistry here at the University of Memphis and the Dean of the College of Arts and Sciences. And I always try to put the acknowledgement slide up front so it doesn't feel like it's an afterthought. All of this work, like I said, is very interdisciplinary and very collaborative. It's really our larger ligand discovery efforts are collaboration between myself, Dr. Perel, and Dr. Judy Cole in biology at the University of Memphis. But we also have a whole slew of graduate students and undergraduates that have worked on these projects as well. Parts of this have been funded by the NIH through an R15 award. Obviously, the University of Memphis Gap Funding is funding the water soluble portion of this. And we've also benefited from the use of instrumentation that's been funded by the College of Arts and Sciences at the University of Memphis. So we're very fortunate in a lot of ways, for sure. So this is really to represent kind of the bigger picture here. So back in pre-2000, I was a postdoc and then a faculty member at the University of Tennessee Health Science Center and studying bioactive lipids like lysophosphatidic acid, sphingosine 1-phosphate, and platelet activating factor. And it was right about that time that it became apparent that these lipids acted through specific cell surface receptors that turned out to be G protein coupled receptors. And that really started a collaboration between myself, Gabor Tiggy at UT Health Science Center, who's in the physiology department, and Abby Perel, who has been in chemistry here at the University of Memphis for the entire time, where we looked at building computational models, doing mutagenesis, studying ligand receptor interactions, and then that morphed into a whole slew of things. What we've been focusing on for the last several years in our extended group is really understudied G protein coupled receptors. And specifically, our real focus and our real goal is on the hardest case for these GPCRs, which are orphan GPCRs, and we'll talk about kind of what that is, what they are, and what the challenges are in a minute. So a lot of what we do is based on using computational models to guide subsequent experimental work. So one of the things that we are not is we are not Pfizer, we're not AstraZeneca, we don't have the resources nor the money to be able to do very large scale, high throughput screening for ligand discovery. And in fact, a lot of big pharma is actually going away from that model. and going more to this kind of a targeted approach in a lot of cases. And the whole goal is to build models that we can use to help prioritize ligand screening. So basically, be able to interrogate very large databases and say, okay, maybe there are 10 million compounds that are available to us. What are the top 100 or maybe the top 50 that we think have the characteristics that we think are important in a specific case? And we do that in a bunch of different ways, and we'll talk about that. And then actually doing the actual experimental work in terms of ligand screening and wholesale assays. And there's a number of ways that we can do that. Now, there are a lot of limitations based on the data that's available and the methodology that's available. And that led us to this idea about alternative ways to express these G protein coupled receptors, and the way that we propose to do it is as water soluble mimics. And again, we'll talk about why that's important. Again, we're taking a computational approach to the initial stages of this to help drive the experimental work. We'll talk about kind of this approach. In larger grant proposals that we've written, we've looked at different kinds of expression systems, bacteria, different bacterial strains, gram-positive, gram-negative, as well as targeted intra or extracellular expression of this protein, and how that would help solve some of the problems inherent in this work. And then we can take advantage if we can express and purify these proteins by directly characterizing them using biophysical methods. And I'll mention that towards the end as well. So a little bit more background on G protein coupled receptors. These are 7 transmembrane, or also called heptahelical receptors. They're embedded in the plasma membrane of cells, and they take cells from signals from outside the cell and transduce that into a signaling cascade within the cell where the ligands don't ever cross the membrane. And some of the situations where these are important, GPCRs are responsible for the senses of smell and vision, as well as signaling of neurotransmitters like epinephrine, dopamine, serotonin, and of course, the reason that we kind of got into this in the first place, bioactive lipids, but also cannabinoids, opioids, etc. So there are lots of inputs into these things, and lots of reasons for interest. So the human genome actually encodes for about 800 total G protein coupled receptors, and about half of those are non-olfactory GPCRs, so they're not involved with smell. So these are the ones that are really important kind of drug targets. As of this month, there are 93 unique GPCRs that have been crystallized and deposited in the protein data bank. And this is a tiny, tiny number when you compare to the number of water-soluble proteins that have been crystallized, their structures solved and deposited in the PDB. Approximately 10% of all GPCR are classified as orphans. Again, we'll talk about that in a minute, what that means and what limitations that puts on work to help elucidate the role of these receptors in normal physiology, normal biology, and pathophysiology, diseases. So it's estimated that somewhere between 33 and 50% of all FDA-approved drugs target GPCRs, and this range of this number really, range in these numbers really depends on how you count them. So it depends on whether you're talking about total number of drugs or sales of drugs and all this kind of stuff. But a lot of FDA-approved drugs target GPCRs. And these are of tremendous interest for general study in the idea about ligand discovery and potentially drug discovery. Our graduate students used to always talk about doing drug discovery, and we had to kind of temper their enthusiasm a bit. We do ligand discovery. Drug discovery is a much more expensive and much more a resource intensive kind of operation for sure. So this is a figure taken from a paper that we published a couple of years ago now, where we looked at that time, 50 representative G protein coupled receptors, and these are all aligned based on their sequence. And what you can see is that this area through the middle here, you have a lot of similarity in terms of structure, and that is based on similarity, high similarity in the amino acid sequence, the primary amino acid sequence. And so this, if we were looking at this receptor in its native environment, there'd be a lipid bilayer here. This is the outer plasma membrane of the cell. This would be all the extracellular space where the ligand would come from and interact in the top about 1/3 or so of this bundle. The ligand binding domain is in there. Down here is the intracellular space where the heterotrimeric G proteins reside. Once a ligand binds, it induces a conformational change that activates these G proteins to go and activate other signaling cascades in the cell. So that's kind of a really simplified topological view of how these things work. So these are actually crystal structures, solid structures. These aren't models at this point. So we have an interest in ligands, and ligands can come in lots of different flavors. Agonists activate receptors, antagonists block agonists from activating receptors, and then there are inverse agonists that actually decrease basal signaling, so that you got lots of inputs in the system, and they correlate with different activity states of the receptor. So these receptors have a range of conformations that they exist in, from active through intermediate and inactive. And that's an important consideration because not all of the crystal structures of G protein coupled receptors are crystallized in the same activation state. We have examples of active, inactive, and intermediate structures. And we may hit on that a little bit as we get through this. So one of the major tools that we use here is homology modeling. And this is an important technique that's used. And the thought process here is that if you have two proteins that have similar amino acid sequence and similar function, then they should have a similar structure. And if you know the structure of one, then you can use that to build the structure of the unknown by analogy. Okay? And the important thing to keep in mind here is that you have to have similarity of amino acid sequence, and you have to have similarity of function to be able to, for this to be a relevant kind of comparison. And we've done some work comparing overall amino acid sequence similarity to a metric that was discussed in this paper from Tony No from UC San Diego, where they looked at a more ligand binding domain specific metric called a coin pocket score. And we've used that a lot in our work over the last couple of years. So orphan GPCRs are some of the most difficult to study GPCRs at this point, because orphans are defined as GPCR without known endogenous ligands. And many orphans lack any known ligands at all, which means that studying their function, what they do normally in a system, what they do under situations of disease or what have you, is very, very difficult because you don't have the kind of pharmacological tools to be able to interrogate these, which is a common practice. So the NIH has a program called Illuminating the Druggable Genome that focuses on GPCRs as well as ion channels and kinases. So basically trying to fund work on druggable targets, right? There are actually 4 levels of classifications in the system, and we focus mostly on these top 2, the T-DARK and the T-BIO, because these are the ones, the examples of GPCRs where the smallest amount of information is known. So T-DARK classification are GPCRs with no known ligands or potencies less than 100 nanomolar. And T-BIO are examples where molecular function is known, but again, there may be a limited number of receptors or ligands, but usually probably have poor potency, but you know, little else is known, and additional reagents and tools would certainly be helpful. And we haven't dipped down into these yet. We've got our hands full dealing with the 40 examples that we've been working with in here, actually more than 40 examples. So our workflow basically breaks down into three main components. The first is the protein modeling, and this really deals with selecting an appropriate template. A related protein with a known structure. And like I said, there are only 93 unique GPCRs deposited in the PDB right now. So in some cases, you have examples or template choices that are closely related to the target that you're interested in, sometimes not. And then we go through a series of sequence alignment and then building the model itself. There's another step that we have to go through in here, which is modeling one of the extracellular loops, which varies. Extracellular loop 2 varies more than the others in both its length and its amino acid composition, and has been shown in a bunch of work, in some cases, to be important for ligand binding. So we want to try to make sure that we get that right. This is just an example pulled from a paper where the green loop in both of these structures, you can see here and here, is ECL2 from the crystal structure. And in this case, using this set of metrics, our purple is the top model, the top scored model. And you can see it doesn't do a very good job under these conditions of recapitulating the experimental structure, but over here it does. So we can do a pretty good job in most cases of predicting what the right answer is. Now, we don't always know what the right answer is, so we have to have ways to be able to pick this out, because we generate thousands of models in each of these cases. Once we have a model to work with, the next step is focused on ligand prioritization. And that's where we actually have many choices of kind of paths to follow. One is this idea about ligand pickpocketing, which is based on work from the CoinPocket paper, which the assumption is that receptors that have closely related CoinPocket scores, right, so that their ligand binding domains are similar, should bind similar things. And this paper was very influential on us. Unfortunately, they just retracted part of it. Fortunately for us, all the modeling bit is still intact, but they had a problem with one of the protein sequences that they used for some of their testing, so that they retracted a bit of that data. But this is still a valid approach in our mind, and we're going full steam ahead to see if we can then be the first to publish a successful pickpocketing example. But then we're also using database mining. So another one of the illuminating the druggable genome tools that's available is the Zinc database, which houses literally millions of compounds that can be subdivided based on drug-like characteristics or commercial availability. We usually deal with a subset of the Zinc database that's about 14 million commercially available compounds. And we do this through a series of either similarity searching to known ligands when that's possible, or through the development of pharmacophores. And pharmacophores are just three-dimensional arrangements of functional groups in space that are correlated to a specific biological activity. And there are two ways that you can generate this. If you have known ligands, you can compare them and figure out what functional groups they have in common in three-dimensional space. When you don't have known ligands, like for example with many of the orphans, we can use a structure-based approach where we can look at the crystal structure of the protein or a homology model derived for that protein, and then use the functional groups in the target itself to generate a pharmacophore to be able to look at these. And we have a couple graduate students. Paige Castleman is revising a manuscript right now on the ligand-based pharmacophore that we should hopefully be resubmitting next week or the week after. And Greg Swabowski is working on the much harder structure-based portion of this. And this is actually broken into two parts as well. But he's working on his first manuscript right now. In fact, he just sent us the most recent draft this morning. And then we can look at ligand docking. We can take those compounds that we think show promise and dock them into our models. And there's a whole bunch of work that we've been doing on this. Brittany Thomas has a manuscript in revision. Mackenzie Griffin is another graduate student who's working on another aspect of that project as well. So this is showing a little bit, just a snapshot of that data that Brittany put together. So what we did is took a bunch of crystal structures, pulled their ligands out, and then tried to dock them back in, either into the exact same structure that they came from or into another closely related structure. So self-docking back into the exact same protein or cross-docking into a closely related protein. And then we subdivided the cross-docking based on either matching the ligand type, so agonist only comparison, or the function of the receptor, or sorry, the state of the receptor, so active, inactive, or intermediate, and the function of the receptor of the ligand, either agonist or antagonist. And what we see is that if we choose the conditions right, we can better recapitulate the easy case of self-docking. Is kind of the short summary of that work. And so the last thing on this part is looking at the workflow for validating our predictions. So the modeling, we always think of as an iterative process. We're taking as much information as we have at a given time. We're making predictions. Then we're going to test those hypotheses. We're going to take the data that results from that and put it back into the system, and then kind of turn the crank one more time. So we've got a number of cell-based signaling assays, and we've got a research scientist that works with the group, Christy Ruddock, who is a machine. She's just been cranking through these things, and she's working on 2 papers right now on these 2 different assays. One of them is a commercial assay that generates light based on cyclic AMP production. Many of these GPCRs signal through this enzyme adenylyl cyclase and either increase or decrease cyclic AMP levels in the cell. And this is some data that she generated using the dopamine D2 receptor, a beautiful dose response curve. The problem is that these assays are very finicky. And they're, in some cases, they're a little more art than they are science. And it's taken us a long time to get these things to be reliable. Sometimes you read someone's protocol in the literature and it doesn't work very well in your hands. So it takes a little messing about to kind of get it to do that. And then this other assay that I'm going to show you here is this nanoluke complementation assay. This is a more non-specific assay. It can look at coupling to many different kinds of G proteins. And this is an assay that was developed by a collaborator of ours now who's in Belgium, Julian Hansen. And so he's helped us set this up in our lab. Again, here's some dopamine D2 receptor data. But again, it takes a long time. You have to transfect in specific receptors into these cells. You have to grow cells. You have to, these are experiments that you have to do consistently. So you're constantly transfecting cells, running assays. It takes a couple of days to do these experiments. And we think that our water-soluble approach may help streamline this a bunch. So basically what we're doing right now in terms of this whole ligand discovery effort is working on a couple of different orphan receptors. GPR88 was the subject of our NIHR15 proposal. GPR52 is another one. We're also working on 40 different TDARC and TBio orphans through our workflow right now. Brittany Thomas has spearheaded that in a big way. And we're using the dopamine D2 receptor, which we'll see a lot when I switch over and talk to the GPCR or the water-soluble stuff, which I'll get to in just a second. Seems to be running a little bit longer than I intended to. but we're doing database mining using the pharmacophores, both the ligand-based and structure-based right now. And one of the big things that we're trying to do is improve our methodology for identifying very good ligand protein docked structures. So we know, based on the way we've designed our experiments, that we have the right answer in these thousands of complexes that we generate, in fact, hundreds at that point. But sometimes it's difficult to pick out, quote unquote, the right answer. So we're working on ways to be able to do that more effectively. So what are the hurdles to this? The single biggest hurdle is the lack of experimentally determined GPCR structures. So only about 22% of these non-olfactory GPCR have available structures. And in that case, and in many cases, it means that we have to use poor template choices for the homology modeling. So templates that have sequence similarities and sequence identities that are much lower than what we'd like to use. And of course, having access to well-curated ligand sets would be great. Having access to money and our partnerships to access screening compounds would be excellent. We've actually been working with an industry partner on some orphan receptors, and unfortunately, we have a non-disclosure agreement signed, so I can't tell you anything about it. It's super exciting. I always feel kind of disingenuous even mentioning that, but we do have an NDA, and I can't talk about it in public. So that leads us to this project, which is really the design, expression, and characterization of water-soluble GPCR constructs. So the whole idea is that we want to be able to take these integral membrane proteins that have this very hydrophobic exterior and express them as water-soluble mimics. And the whole idea is that we could then express these in bacterial systems on large scale, purify them, and then be able to directly analyze ligand interactions using standard techniques, including isothermal titration, calorimetry, et cetera. So the three receptors that we're focusing on right now, and in fact, we've actually pared it down to two that we're going to work on initially. The first one is a human cannabinoid receptor, and these were chosen for lots of reasons, not the least of which is that they have known structures, known crystal structures. And so when we express these and purify them and determine their structure, we've worked with folks at St. Jude before. Like I said, this project started a number of years ago, and we actually gave up on it at one point. We'd submitted grant after grant after grant, and we kept the reviewers kept saying this is an extraordinary idea. It would be paradigm shifting, but we don't think it'll work. And then recently, a group at MIT published a study that basically showed that it does work. So that they have the proof of principle and some chemokine receptors. So CB1, the human cannabinoid 1 receptor, is one target. The dopamine D2 receptor, and you can see these are just snake plots showing the primary amino acid sequence. You can see the difference. The CB1 has a very long amino terminal domain and a kind of medium-sized C-terminal domain, whereas the dopamine receptor has a very long intracellular loop 3, a very short C-terminal domain, and a short N-terminal domain. So there are differences in structures, certainly outside of those membrane-spanning domains. The third receptor that we've proposed here is an ADP receptor that's important in platelets, the P2Y12 receptor. But I'll show you here that it, in a lot of cases, it kind of recapitulates the things that we'll be able to learn with the dopamine D2 receptor. So how do we do the computational work? We took those 50 representative GPCRs and we looked and calculated the outward facing hydrophobic surface areas. And we compared that to a bunch of water soluble globular proteins that are deposited in the protein data bank, and we calculated their outward facing hydrophobic surface areas as well. And what you can see is that the GPCR had a much, much larger hydrophobic surface area than do water soluble proteins. So the whole idea is can we mutate the minimum number of those outward facing residues to more polar residues, maintain the structure of the receptor, and then be able to express it as a water soluble mimic and take advantage of all the tools that we know work for typical water soluble proteins. And we use this QTY code. We actually expanded this to QTYS code, which was used in the MIT work, basically taking more polar residues and replacing outward facing non-polar residues. So again, here's the human CB1 receptor. Here's the amino terminus, which is outside the cell. Here's the carboxy terminus inside the cell. And this is a bit discontinuous just because when we look at these models, we clip off a lot of the fusion partners that they express these with to be able to get them to crystallize. Because these structures have such conformational flexibility, then they do things like add T4 lysozyme to the interior. They replace the third intracellular loop to be able to give a better crystal packing. So all the structures are going to pop up here are in the same orientation. So this is basically representing the computational work that we did. This is the wild type CB1 protein. You can see the green is all the hydrophobic outward facing surface. So lots and lots and lots of hydrophobic areas. And then using those 50 common structures that we had access to at the time, we started looking at common spots and making mutation sets. So if we mutate 28 amino acids, 42 or 65, then this is what the outward facing hydrophobic area looks like. And if we just looked at CB1 and we used our chemical intuition and we just started making directed mutants, and then we could come up with a tailored set. The whole plan for this was really to try to come up with a set that's transferable to as many class A GPCRs as possible. That's where we figure we have the most impact. However, if we have a way to be able to design this and tailor it to specific receptors, that's also a benefit. So what we see here is a plot of the hydrophobic surface area for each of these receptors with these different sets. And what we can see is that the 42, 65, and the tailored really get us down here for the D2 receptor and the P2Y receptor, very near where we need to be in terms of the range that we saw for normal water soluble receptors. And the CB1 is getting us in the ballpark. So we're focusing on CB1 and the D2 receptor first because they will give us some idea about how many changes are really necessary because of the difference in these hydrophobicity surface patches. And so how are we doing this? We're basically generated some plasmids that have a reporter protein. This is the emerald green green fluorescence protein. There's a linker between it, and then we clone our GPCR in with an affinity tag. And this is just a typical bacterial expression vector. The reason that we're using the GFP is because it's been shown previously that this green fluorescence protein will fold properly if the protein before it folds properly. So if this doesn't fold properly, then this will not, and it won't express properly, and we won't get a signal. So it's a way to screen through a whole bunch of conditions quickly. And then the flag tag allows us to do purification later. When we settle on some expression conditions at work, we can just clone in a stop codon here and express only our GPCR. So this was an exciting day when we saw this. Here's a colony of bacteria that's green. Here's one that's less green. So we were super excited when we started seeing bacteria expressing protein where we saw green fluorescence protein being expressed. Super exciting. So COVID has certainly slowed down our experimental progress on this. Right now, all of our constructs, I think we only have one that hasn't been verified. We've definitely seen green colonies noted, and we've done initial protein expression trials that look promising, but we always like to make sure we can verify these things and make sure that they're consistent. I've been in the science game for a long time. I've never been associated with a paper that had to be retracted. I'm very proud of that. So I think I tend to be the tortoise rather than the hare in these things, try to be as methodical as possible. If we can verify protein expression, we can do large scale expression purification and characterize the stability of these things so that we can generate large batches, and we can then pull aliquots out and do experiments where we look at ligand binding, we look at calculate dissociation constants and all these kinds of things. And we've got, we're on track to submit proposals to this NIH Eliminating the Druggable Genome project in July, as well as additional R21 submissions and submission to the National Science Foundation Chemistry of Life program in the fall. I think that is it. I'd be happy to take any questions if anyone has them.
Great. Thank you, Dr. Baker. That was great. I had one question.
Sure.
And it was something you mentioned earlier about big pharma moving away from what's considered traditional R research or R&D and moving to more targeted R&D. Is that because of the success rate being higher with more targeted approaches, or is it just a better way to stretch resources?
So there was a big push for maybe 10, 15 years to generate these really large in-house databases and then just brute force screen that whole library through whatever target you're looking at. And basically that takes huge resources. It takes huge verification, right? You find a hit, then you have to be able to go back and pick that out again. You have to go through all these processes.
Right.
And it's super expensive, and just doing random screening, if I remember right off the top of my head, I think the success rate is like 0.1 or 0.01%.
Yeah. It's something very small, because it's like then
Correct.
When a patent is granted or an application is granted, they have exclusive rights to sell it for something like 15, 20 years, because they need to make all their money back basically for all the money they've thrown into R&D. That's interesting. My wife works in that area, and so it's always interesting to kind of see where research is going. And obviously here at the university, where research is going from an academic perspective.
Well, and we always tell our students, that hey, we can't compete with Pfizer or AstraZeneca from the perspective of those kinds of resources, right? We don't have 27 robots that can do this screening. And we don't have the in-house libraries to be able to do this.
Right.
The other thing is, it's not terribly intellectually satisfying, right? To just blindly screen these things. So the instance where we have some advantage is to be able to use the computational tools to guide what we do experimentally. So a number of years ago, we had a graduate student who did some pharmacophore analysis on a different project, an enzyme that actually makes one of the bioactive lipids that we were interested in. And his initial screening hit rate was 30%. Compare that against the 0.1 or 0.01% success rate from just random screening. We had to talk him down from the ledge, because he was like, oh, I only had 30% success. And we're like, Jeff, you don't understand. Like, it's orders of magnitude better than just random screening, right? So once we kind of put it in perspective, he was much happier with his results.
That sounds great. Great. It looks like there was a question posed, and I'll read it off to you.
Okay.
It's a two-part question. So the first part is, from the first half of the presentation, it seems like it seems like much of the focus has been on the computational end of that project, and not so much on the wet chemistry. Is Christy Reich the only one doing experimentation?
No, we've got because of COVID, it's been more difficult for our graduate students to do in-lab experiments than it has been to do computational work. And so what we've done is try to take advantage of what we can do most effectively and most safely over the last 12 months, which has pushed the computational part of that whole ligand discovery effort much further than what it would have been otherwise. I think we would have had a much more even advance of our progress on the whole ligand discovery, and much more progress on the water soluble construct work had we been able to be in the lab, in a normal fashion for the last 12, 13 months.
Great, great. Thank you. And the second part of that question was, do you think you're reaching a point where there will be a transition towards more wet lab work based on the computations?
Yes, in fact, we're doing that right now. Like I said, Greg has built some structure-based pharmacophores and screened the whole zinc library, and we just did a second round of screening on D2 ligands. And so, I think we talked about Paige also doing some ligand-based work in the same field so that we can use that same D2 construct. So yeah, we're going full steam ahead. And the industry partner that we're working with, we've done a lot of computational work, and they already have the screening. Actually, they've contracted out the screening to another lab, so we don't have to do that work here. And it's a receptor. It's more than one receptor that we don't have assays up and running for, and someone else did. So we're farming that out, and we see that being kind of a model that we want to be able to try to exploit in the future, because we can do all the intellectual part of it, farm out the part that costs a lot of money, and be able to move through these 40 orphan receptors that we're interested in studying much more quickly than we could do ourselves.
All right, great, great. Thank you. The second, actually, that was both of those questions were the first part of his question. The second question then is from the second half of the presentation. The transformations that have been done recently that have gotten everyone excited with the GFP tag in it, who has been doing this work, and has there been any additional verification on the success of the transformations?
So that's a graduate student. That's Christy Dyer. And she's got these constructs all together. They've been sequenced and verified. And we've done a couple of rounds of initial protein expression. And what we're trying to do is work towards large scale production. At that point, we can, we've worked with a group in structural biology at St. Jude before. We actually did, like I said, we did a lot of this work initially on the beta-2 adrenergic receptor, and we actually expressed protein, purified it. We grew crystals, we got X-ray diffraction data. And what we found is that we had contamination from a bacterial chaperone protein. So we learned a lot of important lessons from that about what to look for early in the process. So we're keeping our eyes peeled for those kinds of issues as we move into that. And hopefully we'll have some really exciting results within the next month or two.
Thanks, Dr.
Great,
Baker.
excellent.
Those are my questions.
Yeah. Very cool. If there's no other questions, thank you, Dr. Baker, for speaking today. Our recording will be posted online, and then if there's any follow-up, Dr. Baker, I'll be sure to follow up with you. Thank you so much.
Sounds great. Thank you much.
Thank you.